Hi, welcome to joining me. Let's jump into my computer where I'm going to walk you through how to look at a certificate of analysis. This is actually for MSC cells, very similar to Muse. It's a little different for exosomes. Exosomes has its own thing, and I'll do a video about that later, separate video on that. But this is for MSC cells, and this is a very extensive, this is a 40-page document, and we're going to look at all the variables, all the things that a clinician should look for, and some of the things that, sometimes you're not paying attention to, but are very important when looking at these certificates. So follow me, let's go into my computer right now. Okay, so this is the video where I show you what a manufacturer who's doing the work actually produces. And then at the end, I'm going to show you what the four things that even this certificate doesn't tell you, because the standard is higher than most people in this industry realize. Everything here is education for licensed providers on how to evaluate supply documentation. It is not clinical advice, and nothing in this category is FDA approved. So let's go. Okay, quick reset before we open the document, because cells are not exosomes. Okay, with an exosome preparation, you are counting particles. The whole problem is that counting does not tell you what you counted. With cells, the problem changes shape. You are asking three separate questions, and each one needs its own evidence. One, are they alive? That is viability. Two, are they what the label says? That is identity. And for cells, it means surface markers measured by flow cytometry, not size, not count. And three, do they do anything? That is potency. So here's the sentence I want you holding for the next 20 minutes. Almost every certificate in this category answers the first two questions well, and the third one not at all. And the third one is the only one that has anything to do with whether the product works. Okay, let's go here to page 10. All right, page 10. This is the whole file. Okay, watch, by the way, watch the scroll here. This is 40 pages long. 40 pages long, okay? And there are 9 supporting documents attached behind the cover. Sterility report, endotoxin report, mycoplasma report, donor serological report, the COFA, the trip LE COFA, PBS COFA, prime, this is the freezer, the freezing agent, then donor serological report, then certificates of analysis for the attachment reagent, the wash buffer, the cryopreservation medium, and the isolation enzyme, and a clean room certificate. Here's the concept of real certificate of analysis. It has children. The cover page is a summary of evidence, and the evidence is supposed to be attached, like in this case. When a supplier sends you a cover page by itself, they have sent you the abstract and kept the paper. So here's your first habit, and you can use it on any document anyone hands you. Pick one number at the front of the page at random, follow it. If you can trace it to a lab report with a method, a date, and a signature, the package is real. If it dead ends on the front page, you are holding a summary, not a certificate. Okay, so there here, all the certificates, just showing it to you that they're here on page 11, 13, 15. All the certificates are attached to the summary on the first page. Okay, top of page 1. Okay, let's read it slowly with me. 25 million umbilical cord mesenchymal stromal cells. 1.75 milliliters cryopreserved. Passage 4. You can see here, passage 4. Single female donor umbilical cord stored at or below minus 140 here degrees Celsius. Right here at the top right. Manufactured October 25, used by October 27, right here. Used by date of manufacture, and use by date right here. Now do this with me because it takes 8 seconds and almost nobody does it. Okay, so here we go. Cell count. The certificate says 1.4 times 10 to the 7th cells per ml. Multiply by 1.75 ml and 24.5 million. So the label says 25. That's close enough. That's pretty good. That's internally consistent. It took 8 seconds. You do the math over here. You see this number over here? Okay, that's the habit. Anytime a certificate gives you a concentration in the volume, right, multiply them and check that you get the number on the front of the vial. Most of the time you will. The times you do not are exactly the times you need to check it. Okay, passage 4. That number is there right here because cells change as you expand them. A certificate that names the passage is telling you the manufacturer controls for it. Your question back is, what is the upper passage limit and what data sets that limit? Storage conditions right here, minus 140 Celsius, that is the vapor phase liquid nitrogen, not a freezer. Practically a clinic that cannot store. There is depending entirely on the shipper. So ask about the shipper the whole time and whether there's a temperature logger in the box. Okay, let's go to page 6, the donor section. And this is where this package pulls away from the field. You know, a weak donor section reads like this. Donor screened, no high risk findings, serology negative. That's a conclusion with nothing behind it. Watch what this one does instead. So we can go to, let's go to page 15. Page 15. You see the final report on the donor and product testing. So a donor identified that ties to an attached third-party report, an accredited tissue bank as the ordering organization, a named reference laboratory, and then every single assay listed individually with its own timestamp and its own result. See, hepatitis here, hepatitis B core total, date completed, hepatitis B surface, AG automated, non-reactive, date completed, hepatitis C, HIV antigen and antibody, HTLV, syphilis, nucleic acid testing for HIV, hepatitis B, hepatitis C, and West Nile virus. Here, West Nile virus, non-reactive. Okay, so let's scroll down, same page. Okay, and you see the test reference section on page 16, the following page. It has, page 16 goes even further. Every assay gets its methodology written out by name, by platform, by detection principle. This is a level of transparency you will rarely see. See right here? Every assay has a methodology description. The 3 questions to ask any supplier about donor screening. Which assays named individually, not summarized, who ran them, and are they CLIA certified? Okay. And what was the interval between tissue recovery and the donor blood draw? That third one matters more than it sounds, because a donor test results, a donor test result only means something if it reflects the donor's status at the time the tissue was actually taken. Okay, so let's compare some of these dates. So let's use it right here. Page 1 says, here, let's go back to page 1. Page 1 says the tissue was obtained on the 23rd of May. You can see right here, date of birth, tissue donation, May 23rd. Okay, now let's go to page 15. 15, the collection column here, page 15 says the donor blood was collected on the 14th of May. That is 9 days earlier. 9 days may sound like a problem. Here, right here, 14th of May at the bottom, you see. That is 9 days earlier. 9 days may sound like a problem, but page 18 is a tissue swab collected on the 13th of May. So let's go to page 18. You have a tissue swab. Yes, that was actually collected on the 15th of May. Okay, no, 13th of May, right here. The sample type, Tesco dual swab, 13th of May 2025, Central Standard Time, which, you know, this suggests the tissue was actually recovered on the 13th, and that the date on page 1 is when the manufacturer received it, in which case the blood draw was 1 day after the recovery. That's completely fine. So which is it? I do not know. That's the point. You know, 2 readings, one is fine, one is not, and the document doesn't tell you which. But this is not me catching anybody out. This is me showing you that one word, recovered versus received, is the difference between a document that answers the question and one that leaves it open. Ask, just in case, ask. Let's go to page 5. This is the diagram. This is the page that most people skip, and it might be the most useful one in the file. You see here, you see there are 6 stages: sourcing, isolation and expansion, harvesting, final formulation, cryopreservation, lot, testing. Here's why this matters more than any single result in the certificate. The process tells you what was done, and that's what lets you judge whether the test results actually mean anything. You know, a sterility pass, you know, on a product made in an uncontrolled room is not the same finding as a sterility pass on a product made in a classified clean room. The second one had systems designed to make contamination unlikely in the first place. Okay, xeno-free reagents. Right here, xeno-free recombinant collagenase passage 1 to 4, isolation in a classified biosafety, biosafety cabinet, expansion under low oxygen, passage range stated 1 through 4, controlled rate freezing, not a straight freeze, and quarantine until every safety result comes back. Okay, let's go to the bottom of page 6. So this is the manufacturing summary, production, quality control. So you have here, you see page 6 and 7 expand every one of these boxes into a paragraph and name the reagents by manufacturer. So you have the isolation reagent, detachment harvest reagent, production reagent, details. You know, now the test. Take any claim on these pages and ask what document supports it. What documents support the claims that we see here? So if we go to page 19, xeno-free reagents. There are 4 reagent certificates attached. Page 19, page 21, page 23, and page 38. Classified clean room, there's an ISO certificate on page 40. ISO certificate on page 40, very important. That is a supported page. So a process flows with nothing behind it is a marketing graphic that happens to be drawn in boxes. Okay, so let's go now to page 3. Let's go up there. This is the centerpiece. Nobody teaches this to clinicians, and it's not hard. You know, first, the standard. The International Society for Cell and Gene Therapy publishes minimal criteria for what counts as a mesenchymal stromal cell. 3 parts. 1, the cells stick to plastic in standard culture. 2 surface markers, at least 95% positive for CD105, CD90, and CD73, and no more than 2% positive for CD45, CD35, CD14, or CD11B. No, CD19 and HLA-DR. 3, they differentiate into bone, fat, and cartilage in the dish. Remember, 3, we're coming back to it. So let's look at these panels. Page 3, 9 panels. Let me teach you the axis once. The horizontal axis right here is fluorescence intensity on a log scale. So further right means the more of the marker on the cell surface. Vertical axis is percent of maximum, which is just a normalized way of showing how many cells sit at each intensity. The vertical line is the gate. See, the vertical line is the gate, and the percentage printed on each panel is the fraction of cells falling to the right of it. So in this case, you have 99.348%, 99.348%, 99.259%, 99.425% falling to the right of this line. That's the whole skill. Now just go in and read them. So CD, for example, CD105, the first one here, CD105, 99.3, CD90, 99.2, like CD73, like I said, 99.4. So against a 95%, against a 95% floor. Okay, so the middle row, let's do the middle row, HLA-DR 0.11, CD19 0.548, 0.6, the CD45 0.61, CD11B 0.35, CD34 0.178 against a 2% ceiling. Comfortably inside the standard on every single panel. That is a well-run identity test. Now look at the 9th panel, this one right here, bottom right. 8 of these have antibodies on them, this one has nothing. It is the unsustained control. It reads 0.049%. That panel is the most important one on this page, and it's the one everybody skips. Here's why it matters, because those gates, they are drawn by a human being. If somebody puts the line in the wrong place, the percentages come out whatever they want they come out to come out. The unsustained control is the proof the gate is set correctly, because it shows you what the instrument reads when there is genuinely nothing there, essentially 0. That one panel validates every other number on this page. So if a flow report ever reaches you with no unsustained control and no isotype control, the percentages on it do not mean anything, and you are entitled to say so. One more. Here, the summary box against the data. Okay, the summary at the bottom of the page 3 lists the negative markers as CD14, CD19, CD34, CD45, and HLA-DR. Now go back up to those panels on the same page. The one that actually ran is CD11B, not CD14, it's CD11B. The standard alone, the standard allows either one, so nothing's wrong with the sentence here, but the summary does not match the panel. So, and the reason to catch that is simple. Summaries get copied into sales decks, and data does not. So read them against each other every time. And ask one more question. Who ran this? In-house flow and third-party flow are different levels of evidence. Neither answer disqualifies anybody, but the answer tells you how the manufacturer thinks about verification. Okay, let's go to page 1. Let's go back to page 1. Page 1 gives two viability numbers, okay? And they mean different things. Production viability, 98 to 100%. That's measured while the cells are growing before freezing. It tells you the culture was healthy. Post-thaw viability, against a 93% spec. That is measured after freezing and thawing. Okay, so let's go to page 2. Let's look at the paragraph. Let's look at the paragraph under the micrograph. So page 2 gives you the actual post-thaw result. 93 to 96% right here, post-thaw viability. So that second number is the one that matters because that is the condition the product is in when it reaches a patient. So if a supplier quotes you one viability number and does not say which one it is, assume it is the flattering one and ask for the other. Okay? So 2 follow-ups and it is reported as a range. So ask how many vials were tested and what each one read. Let's go to page 8. There's a section here on the cryopreservation. Ask when it was measured. Page 8 tells you 72 hours after freezing. That validates the freezing process. Hold that thought because it does not validate anything else. So back to page 2. The hard part. Okay, the last line of page 2, right here, says post-thaw viability ensures the cells will retain sufficient potency. That sentence is doing more work than the data supports. So viability means the cell is alive. Potency means the cell does something. Those are 2 different claims, and nothing in this package bridges them. A cell can be perfectly alive and functionally exhausted. This is the simple, most common conflation in the entire cell therapy market. And now you will hear it every time somebody makes it. So let's go to page 4. 3 safety tests. Page 4 tells you the methods, and there is a lesson buried in each one of them. Sterility, no growth, run at a named contract laboratory on an automated culture system. Compare with P9, second line. Here are the tests. Okay. Small note. Okay, page 4 describes a rapid respiration method. So page 9 describes a 14-day inoculation. Both are legitimate methods. They are not the same test. That is worth 1 email, nothing to be alarmed about. So let's go to page 14. Mycoplasma. Negative from a specialist lab. Direct culture plus an indicator cell culture assay. Tests started on December 22nd, completion on January 19th. 28 days apart. Why anyone cares? Mycoplasma contaminates cell cultures invisibly. It doesn't cloud the medium and it walks straight through a standard sterility test. Any human cell product should carry a mycoplasma result. If a certificate doesn't have one, that is a real gap. So always look for the cell culture mycoplasma test. Okay, so let's go to page 12 for a second here. Now the endotoxin, and pay attention to this because this one flips. Page 1 and page 12 both say less than 1.00 endotoxin units per ml. Okay, that looks like a limit, not a measurement. Okay, and limits without values are just weak reporting. So watch what happens when you read page 13 instead of the summary. Okay, so cartridge range right here. Let me find it. Cartridge range right here. 1 to 0.01. So the instrument resolves down to 0.01. Dilution 1 to 100. Here, right here. So 0.01 times 100 equals 1.00. The floor is a function of the dilution. It's not a property of the product. So look at the sample data table. Both channels here, you go up, scroll up a little bit. Both channels reaction time greater than 890 seconds. That means they ran past the end of the detection window with no reaction at all. And the spike recovery, bottom right, inside the 50 to 200 percentile range, acceptance range, right here, spike recovery range, 50 to 200 acceptance range, which proves the dilution was not hiding interference. Now, 2 lines of arithmetic. Under 1 endotoxin unit per ml, in a 1.75 ml dose is under 1.75 units in the vial. For a 70 kilogram patient, that is about 0.025 units per kilogram against the commonly used threshold of 5. There is roughly a 200-fold margin. So a number that looked like a weak disclosure is actually an excellent result. And you can only tell by opening the attachment and doing 2 lines of math. So that cuts both ways, and that's the honest lesson here. Sometimes reading the raw data makes a certificate look worse. Sometimes it makes it look, considerably better. You can read it either way. So let's jump back to page 1. Last thing in this segment, look at the calendar. Page 1, manufactured October 24th. Page 14. Final mycoplasma result, January 19th. 87 days. And page 9 states the batch stays in quarantine. Here we go back to page 9. You'll see that the batch is held in quarantine until every test comes back negative. Understand what that means commercially. A manufacturer running a 28 days test and holding the batch is choosing to sit on inventory for 3 months. That costs them real money. So when somebody offers you same week availability on a cell product, the question is, what testing was completed before it shipped? Here, 11. Notice one more thing across all of these reports. Everyone names the laboratory. Everyone names the laboratory. Gives an address. Carries an accession number. That is independently verifiable. A certificate that says tested by an accredited third-party laboratory without naming it is not verifiable. And unverifiable is functionally the same as unstated. Okay? And coming to the end here, let's go to page 1. Look at the cryoprotectant roll right here. So providers, they think about the active ingredient, but look at what is actually in the vial. Page 1 names the cryoprotectant. Page 8 tells you the formulation. Let's go to page 8 just to make sure it's there. It tells you the formulation of the cryopreservation. 25 million cells in 1.75 milliliters of cryopreservation medium. Okay? So that medium is not a manufacturing aid that gets washed out. It is the majority of the injected volume. So you go, let's go to 19, page 19, here, the page 21, page 23, finally page 38. Every reagent that touches this process has its certificate attached for every single reagent. Detachment reagent, wash buffer, cryopreservation medium, isolation enzyme. The first two get washed out. The third one doesn't. So the ask is simple. What is in the vial besides the cells? Right? What is in the vial besides the cells? And can I see its certificate of analysis? Here the answer is yes, for every reagent in the process. And now the thing a sharp medical director will find, so you should find it first. Read the banner at the top of the pages 19 and 21. This is page 19, where we are research use or manufacturing use. Okay, not intended for direct administration into human or animals, right here. Okay, now page 28, bottom left. There we go. Let's go to page 28, bottom left. Same language for the cryopreservation medium. Research or manufacturing base use only, not for injection. Okay, now page 1, top of the row, here we go. Here's a product table, and read the very top line of page 1. The manufacturer has written the same designation on their own certificate. That designation is generally common across this field for materials of this class. And this package includes a full toxicology study of the cryo medium, pages 24 to 28, which is the appropriate supporting document to hand over. I'm not telling you what to conclude from that. I am telling you it is written on documents you were given that you should read before somebody in your practice asks you about it, and that the right position is having the supporting data in hand instead of being surprised. Everything I've shown you for the last 20 minutes is generally good work, and it still doesn't answer 4 questions. 1, potency. Go back to those ISCT criteria. Adherence, documented on page 5. Phenotype, let's go page 5. Phenotype, documented beautifully on page 3. Differentiation, nowhere in these 40 pages, and no other functional assay either. So 2 out of 3. Nothing here demonstrates the cells do anything. So 2, what supports the shelf life? Page 1 says use it by October 27, a 24-month dating. So the post-thaw viability behind it, page 8, was measured at 72 hours. Ask for 6, let me look at page here, page 8, was measured at 72 hours, after 72 hours. So ask for 6, 12, and 24 months time points. Okay. So 3, genetic stability. Page 1 says passage 4. Ask for karyotype and or another genetic stability assessment, and ask for population doublings. 4, there is no lot release date anywhere on page 1. We can infer from page 14 that the testing finished in mid-January, so release cannot have come before that. But the date itself is missing. And a release date with a quality signature is the line that says a human being reviewed the whole file and authorized it. 4 gaps in the best package I have read in this category. That is not a knock on anyone. That's the standard being higher than the market currently delivers. If you take one thing from this series, take that. And if you want to reach out to us, you want us to take a look at a redacted certificate, please book a call, and we'll be happy to discuss. and discuss other biologics that we can help your clinic with and other programs that we have here at MSC Exos. We are the operational system for clinics in longevity in regenerative medicine. Thank you for joining me. I hope this was helpful. Goodbye.